welcome back from your break. I hope your brains are now fresh and ready to absorb more plant anatomy. We are going to start by looking at the structure of a stem. We've already revised the different types of tissues, so we're going to look at what types of tissues we find in a stem, what they do there, and how they are arranged. What tissues will be needed in a stem? Here is your stem. Let's think. Does it need meristematic tissue? Will it need to grow? Does it need epidermal tissue? And if so, which ones? Would we find parenchyma, collenchyma, sclerenchyma, and xylem, and phloem? And if so, what do these cells actually do? We would find meristematic cells in buds. That's a bud. And that is where new leaves and flowers grow. We would also find epidermal cells, and quite important, they would cover the outside of the stem, and they would need a cuticle to prevent water loss, plus they would need stomata to get in the gases they need. Would we need parenchyma? Most definitely, to fill spaces and store food. Then, our supporting tissues. Collenchyma. Are we going to need collenchyma? Very definitely. That is what keeps the stem standing upright. Some plants only have collenchyma for support. Others also have sclerenchyma. And remember, sclerenchyma is much, much tougher, so it does a better job of support. Then, because one of the functions of the stem is to take water upwards and food downwards, we would find xylem, which is our water transport tissue, and phloem, which is our food transport tissue. So these are the cells that we're going to find in a stem. The next question is, well, where in a stem will we actually find all these tissues? This is a cross section through a stem. In other words, we've taken a stem and we've cut it across. And if you look at it, it's very, very confusing to start. But let's start with the easy things, the things that are easy to recognize. And obviously, the first one that's easy to recognize is, does it have a skin? And the skin is the epidermis. Then there are a whole lot of big cells in the center, and if you look at them, they've got thin walls. What type of cells would those be? Those would be parenchyma cells. Large cells, thin walls with air spaces between them. Then the rest all looks pretty confusing. How can we make sense of it? Remember, the darker the color, so if we look there, the color is quite dark. That shows that the cells have got thick walls. There, the color is also dark, so those are cells with thick walls. If we look here, these cells also have thick walls. And if we have a closer look, we're going to be able to identify which of the cells with thick walls they actually are. If we look at the next picture, this looks kind of different and sort of the same to the previous one. And the reason is that the two are sections of different stems. So every stem would look slightly different, but if you look, there is the same pattern. And that's what we're looking for, the pattern for the layout of these different tissues. Another thing, the color is different. Look at that color. And if we look at this picture, look at that color. And the reason why the colors are different is simply because these two pictures have been stained by different dyes. Because most of the cell walls and things like that in a plant are colorless, so we need to stain them so we can see them clearly. And the different colors actually simply are the different stains that have been used to bring up the different types of tissues. So what we're going to look at now is we've already found the epidermis. Sure, that was the easy one. And we have already found the parenchyma cells in the center, and that area of parenchyma cells is called the pith. Then we were a little bit confused by all the different cells with thick walls. Now, if we look at that pattern, you see there are a whole lot of those, and those are all vascular bundles. You can see all the way around near the outside, there are a whole lot of these vascular bundles, and that is where the xylem and phloem is found. Then, just underneath the epidermis, the cells there you can see are it's quite dark, meaning the cells have got thick walls, and that is your collenchyma, and that makes sense because the collenchyma is there for support. So we have identified collenchyma. So let's look at the next picture, and that puts in words the fact that there's collenchyma under the epidermis, there's vascular tissue, they're called vascular bundles, and those are the little patches of xylem and phloem, and then the center of the stem with the big parenchyma cells, that is called the pith. I know it's very confusing looking at all these cells everywhere. So what we're going to do now is we're going to look at a simpler diagram which simply shows us where the different types of tissues are found, and hopefully that will make it a bit easier for you to understand the pattern we find in the stem of a plant. This is a section across a plant, and that bit there is the cross section. You note there's no detail of the cells. It's simply showing us where the different types of cells are found. And something like that is one of the vascular bundles of xylem and phloem. Here, the section has been cut downwards. So here you are looking at the xylem vessels from the side. So this is called a longitudinal or lengthways section. Then, looking at the cell from the outside is called a side view. So this diagram shows us three different views of the arrangement of tissues in a stem. The part that is most confusing is these vascular bundle things. So what we're going to look at now is we're going to up the magnification of the microscope and look at only part of a stem, but it means we can see the vascular bundle in quite a bit of detail, which makes it much clearer and much easier to work out what is going on. This circled area is the vascular bundle. You can probably pick up, there's the epidermis, there's the collenchyma, but here we're focusing on the vascular bundle. We can see here nice big empty cells, and those would be your xylem cells. They've also got thick walls. Then what you see here are smaller cells which also have thick walls. And their walls are evenly thickened, so that is the sclerenchyma. And between the two are thin-walled cells. And there's two different types of thin-walled cells here, filling that space of thin-walled cells. That is the phloem. And you can pick up there are little companion cells between the big sieve tube cells. Then below that is a layer of flattened elongated cells, which we haven't come across before. Those are meristematic cells, and they form what we call the cambium, and that is what enables a plant, as it gets bigger, to grow more xylem and phloem, because if it's bigger, it's going to need to transport more water upwards because it's going to have more leaves. This is simply another picture. And if we look at it, there are the fibers. Why do they look so different? A different dye has been used so they show up as a different color. There's the phloem with the big sieve tube cells and the small companion cells with thin walls. There's the vascular cambium, the flattened cells, and here we can find our nice big xylem cells. It's often easier to understand by drawing a plan or a line diagram of a stem. And a line diagram simply shows where the different types of cells are found, doesn't actually tell you what the cells look like. So let's have a look at this diagram of a plan and see if we can recognize all the different regions. Often it's easier to look at a plan diagram which shows you where the different types of cells are found without actually giving you any detail of what the cells look like. This diagram is a simplified diagram showing vascular bundles inside the plant stem. Let's now have a look and see what can we identify from what we have already talked about. First one is these things here, okay? And these, at the tip of the vascular bundles, those are patches of xylem. What else do we find in our vascular bundles? We find phloem, and that's where the phloem is found. So that would be an area of phloem. Then we would also find the bundle cap of sclerenchyma. The last part of the vascular bundle is this area here between the xylem and the phloem, and that is where the cambium or meristematic tissue is found. So we've now looked at the detail of the vascular bundle. We're now going to look at some of the other parts that we can recognize. Easiest one to recognize always is the epidermis, because every plant part has to have an epidermis. Also, every plant part will have cells filling in spaces, and those would be parenchyma cells, and the parenchyma cells are found in the middle with the pith. This particular diagram doesn't show the collenchyma. Next picture we're going on to is a drawn diagram showing the detail of a vascular bundle, and let's see if we can pick up the different parts from this. Easiest one is always xylem. Very easy to pick up, large cells with extra thick walls. Next easiest one to pick up is going to be your phloem, and we can pick that up because there are some big cells, which are the sieve tube cells, surrounded by smaller cells, which are the companion cells. Another easy one to identify is this part here, which is the bundle cap, and that has got cells with thick walls, but they are small cells. You can see the difference between the size of a xylem cell and the size of one of those sclerenchyma cells. That's the way you can tell the difference, because they both have thick walls. Then we can also find the other parts of the stem, and this one shows very, very nicely. Here we have the epidermis, and this also shows very nicely the collenchyma with the thickened corners. It also shows very nicely a whole lot of parenchyma cells between the vascular bundles and filling the center. The parenchyma cells that are between the vascular bundles form part of the cortex, whereas those right in the center form part of the pith. There's really not much difference in their structure, it's just where they are found.